Hello bachcho, the question is the test tube baby program employs which one of the following techniques? So first option is intra cytoplasmic sperm injection, second is intrauterine insemination, third is gamete intra-fallopian transfer, and option D is zygote intra-fallopian transfer. So test tube baby program. So test tube baby, actually the baby is not born in test tube. Actually the fertilization is done in test tube in laboratory under controlled condition. So this technique of formation of test tube baby is called the in vitro fertilization, IVF. So here these some techniques are given, and these techniques are together known as ARTs or assisted reproductive technologies, because these technologies assist the infertile couple to have a child. Now there are several kinds of STDs, like first is ICSI or Intra Cytoplasmic Sperm Injection. Here the fertilization is done in laboratory, means in vitro. What is to be done? That ovum is collected from the female. Now this ovum is actually at the secondary oocyte stage, so it is collected directly from the ovary, or it can be collected from the fallopian tube when it is ovulated. So now the secondary oocyte has a nucleus, so this is the nucleus, and cytoplasm. This is the cytoplasm of secondary oocyte. Now what is to be done? That sperm or semen is to be collected from the husband or the donor, and this semen through injection is transferred directly into the, here the semen is transferred directly into the cytoplasm. So it increases the chances of fertilization. So the chances of fertilizations are very high. So here the zygote will be formed. So this technique where the semen or sperm is directly injected into the cytoplasm of the egg, so this is called the ICSI or intracytoplasmic sperm injection. So here we can see that fertilization is taking place outside the mother's body in laboratory. So this baby formed will be the test tube baby. Now this zygote or embryo will be transferred into the female, either in the fallopian tube if it is below 8 blastomere stage, or it will be transferred into the uterus if the embryo is above 8 blastomere stage. So this baby, actually it will develop inside mother's uterus, but the fertilization is taking place in laboratory in test tube. So that's why this baby is called test tube baby. So we can see that in intracytoplasmic sperm injection, the fertilization is in vitro. So the baby is said to be a test tube baby. So it is our right answer. So the option A is the right answer. But if we look at other three options also, then the other one is intrauterine insemination. So intrauterine insemination or IUI is the method of insemination or artificial insemination, where the semen is transferred deliberately into the uterus of a female. So here if the male is unable to produce the sperm, so another male's semen is collected and transferred into the uterus of the female, or if the husband is having any problem during the sexual intercourse, so also in that case artificial insemination is to be done. So here the semen is collected and it is inserted into the female by a doctor in her uterus or fallopian tube or vagina or cervix. So here the IUI involves the insemination of semen or the insemination in the uterus of a female. So here the fertilization will occur in the mother's body, so that baby is not said to be the test tube baby. So this is incorrect. The third option is GIFT or gamete intra-fallopian tube. Here what happens? The ovum is collected from a donor female, and this ovum is introduced into the fallopian tube of another female who is unable to produce the ovum. So this technique where the gamete or ovum is transferred from a female to another female, this technique is called gamete intra-fallopian transfer. Here also the fertilization will be inside the mother's body, so this is the case of in vivo fertilization. So here no test tube baby is formed. So this is discarded. Now the next option is zygote intrafallopian transfer. So here I told you that zygote or embryo up to the 8-celled stage is transferred into the fallopian tube. So this transfer is called zygote intrafallopian transfer. So this is a technique of embryo transfer. Here the means zygote can be formed into another donor female, or it can be transferred into another female. So here there is no case like in vitro fertilization is required. In case of in vivo fertilization, the ZIFT can be performed. So this one is also incorrect. So the right answer of this question is option A. I hope this explanation is very clear. All the best.